Welcome to the Fatty Acid Forum, sponsored by Virtus Nutrition. Join Dr. Barry Bradford from Kansas State University as he explores how nutrient metabolism influences the immune system of a dairy cow. Now, here's Dr. Bradford.
In the last presentation, we talked a little bit about how supply of potentially deficient nutrients might influence immune function, but the interactions of nutrient metabolism and immunity are also very important and be less obvious. So I want to go through that a little bit in this presentation. So today we're going to focus on this link between metabolic health and immune function right here. So first of all, some just simple evidence that there is an important link here. On the left-hand side, you can see data from a 1989 study which was surveying cows that had problems in early lactation, including metritis or acute or chronic mastitis. And what these scientists found is that cows that had these conditions early in lactation then had anywhere between a 1.5 to 2.5 fold increase in their subsequent risk of ketosis. So in other words, cows that had these infectious disorders like metritis and mastitis are also then later at greater risk of having metabolic disease. Okay, and then if we look at the flip side of the coin based on a couple of studies over the course of several years, on the right-hand side, this shows that cows that already had ketosis, so that started with metabolic disease, were then subsequently at greater risk of both metritis and mastitis, and those were of pretty high magnitude. So with the metritis study, it's about a 2.5-fold increase of risk of that disease following ketosis, and about a 3.5-fold increased risk of mastitis after ketosis. It's true both that infections increase the risk of metabolic disease and that metabolic disease increase the risk for infections. So you can see how this can turn into a sort of a disastrous scenario for some of the cows out there. So what are some of the mechanisms underlying this? Well, we actually have some decent evidence of this. One nice thing about immune function is that you can remove cells from the body and study them in vitro in a way that's much more controlled than you could possibly accomplish in a cow that's sick and not eating well and has all sorts of mixed problems. So in this study, cells were removed from cows, and these neutrophils were evaluated for their ability to respond to pathogens. And two of the things that are very important for neutrophils to do is to engulf pathogens that they encounter. Okay, and what they found in this study was that as ketone body concentrations increase, this is BHBA, from 0 to 8 millimole per liter, millimolar, the ability of these neutrophils to engulf bacteria decreased dramatically, going from about 90% of those cells doing that to only about 20%. Now, once they had them engulfed, the cells remained able to kill the bacteria just fine, but the point is if only 20% of the cells are engulfing the microbes in the first place, not a lot are going to get killed on the whole. There's another way that neutrophils can kill pathogens, and that's called extracellular killing, and I'll show you an image of that in the next slide. But the amazing thing is this process, the other tool that is in its arsenal, was also dramatically suppressed as ketone body concentrations increased, going from, again, about a 55% of the cells doing this function to only about 5% at the 8 millimolar BHBA concentration. And the range from subclinical to clinical ketosis is somewhere in this range. So these are not crazy high concentrations that are meaningless. These are relevant in an early lactation cow. And this picture just shows in kind of a neat way what it looks like when these neutrophils use their extracellular killing ability. So they form these extracellular traps, they're called, by essentially committing suicide and turning their DNA into sort of a spider web that is then released from the cell and forms a complex with proteins that we think entraps and essentially leads to the death of pathogens. Okay, so that study is interesting, and I think it's relevant, but it's still in vitro, so there's only so far that you can take it in terms of interpreting inside the whole animal. In my group, we've done some studies lately where we have simulated the inflammation that accompanies a low-grade infection. So what we're trying to do is not simulate an acute septic mastitis case that a cow is in terrible shape and is obviously in trouble. We're trying to mimic the sort of low-grade inflammation that might accompany subclinical metritis or subclinical mastitis. And so in this study, we started cows within a day after calving, and for the first 7 days of lactation, every day we gave these cows a low-dose treatment with tumor necrosis factor alpha, called TNF. And at this low dose, we did not observe any increase in body temperature, and yet this mild inflammatory state caused cows to eat about 15% less feed. And as you can see here, the incidence of ketosis, although this wasn't statistically significant given the small groups that we had, in the control group that was not given an inflammatory agent, only 9% of the cows had subclinical ketosis, but in the two treated groups, that rose to 27%. And the effect on dry matter intake was significant. And as you know, the decrease in feed intake in early lactation is a very clear risk factor for ketosis. So this kind of lends itself to the story that cows, even with mild infectious conditions, might be at greater risk for metabolic disease, if for no other reason than the fact that they may not eat as well. And so if you put a group of studies together that kind of show these same types of responses, we've come up with a model that we think can explain at least some of the cows that really get derailed in early lactation. And it doesn't always start with excessive lipid stores, but this is a key risk factor for transition problems, and so it makes sense that this is part of the story. We think that excessive supply of fatty acids to the liver can directly alter metabolism in the liver, probably in a negative way, but also because it leads to high ketone body concentrations, can cause problems on the immune function side, because as I showed you a minute ago, neutrophils and other studies have shown that macrophages as well are heavily impacted by high ketone body concentrations, as well as NEPA concentrations for that matter. So having a suppressed immune system puts the cow at greater risk to have a clinical or subclinical infection event. That then leads to a spike in inflammatory agents like cytokines. And here's where the vicious cycle comes in, because these cytokines can actually travel through the bloodstream, reach the liver, and then directly alter metabolism. And it appears that they, in some cases at least, can enhance ketone body production and kind of continue this cycle. So if that's true, then it should also be true that some unexpected benefits would come about by derailing some of these problems. So on one side, we might expect that doing something to improve metabolic function might actually increase immune function as well and prevent infectious disorders. So let's look at one study that kind of supports that view. In this study, cows were fed 15 grams of choline daily from 25 days before calving through the first 80 days of lactation. And this is a study published in 2012. Now, choline is typically fed as an agent to help decrease liver fat content, which should improve liver function, and therefore the logic is that you should prevent metabolic diseases. And true enough, in this study they found a significant decrease in ketosis rates. Okay, so far this looks fairly predictable. But then what they found that was maybe less predictable was a tendency for a decrease in mastitis incidence. And then in terms of total morbidity, including these two conditions and everything else, total morbidity was very, very significantly decreased. So the p-value of less than 0.001, going from nearly a 60% incidence of some kind of problem in early lactation down to 40%. And I don't think you could explain that simply by direct effects on metabolic function without having some trickle-down effects, if you will, on immune function and other capacities of the body. So one of the stranger concepts that's come about in the last few years is the idea that maybe some nutritional deficiencies can be addressed in ways other than through the diet. And so John LaPaulis and others at the NADC in Ames, Iowa, part of the USDA system, has been studying the impact of vitamin D on both isolated immune cells and on mammary gland immune defenses after mastitis. So in this case, cows were challenged with strep uveris, and the response to that simulated infection was monitored. In this study, you can see here that the control animals have a dramatic rise in bacterial counts of strep uveris, so it's obviously proliferating. But in these animals that received just a small dose of 25-hydroxy vitamin D3, there was about a 4 log decrease in the concentration of this pathogen in the 2 to 3 days after this treatment was carried out. And probably even more dramatic was the response in somatic cells. So this is obviously a pretty blunt instrument to assess mastitis response, but you can see here that the cows given the vitamin D really show no rise at all in somatic cell count until about the 8th or 9th milking, whereas the control animals spiked to about 1.4 million cells per mil just 2 days after the infection was induced. So a really dramatic response here, and this is consistent with work on isolated cells showing that vitamin D in the cells themselves can actually improve their ability to respond to pathogens. So overall, I think beyond simply addressing nutrient deficiencies, we can also think about supporting metabolic health in general to help improve immunity. And so that might mean supplying nutrients or bioactive ingredients that alter function even if those nutrients are not necessarily deficient. On the other hand, if we can prevent infections by doing a better job with our parlor routines or with stall preparations, that can actually potentially help prevent metabolic disorders, because by preventing infection, we can keep cows eating, we can prevent metabolic disorders, and maybe prevent direct problems with inflammation as well. Although this is kind of the early stage of this kind of research, but we should think about sometimes the fact that just because a nutrient is deficient in a certain location in the body doesn't necessarily mean we can address that deficiency through the diet, because the trafficking of those nutrients is regulated by the body and not always in the way that we'd want it to be. So milk is relatively low in vitamin D, and that is partially why we think that supplementing vitamin D through the teat canal might actually influence immune function in the gland. Okay, so overall there's a lot of potential here to better understand and improve immune function of cows through supporting metabolic health.